Hello friends, welcome back to my YouTube channel Nasrin Padasalis. In this video, I'm going to discuss the topic from which question has been asked regarding to join CSIR June June 2022. So in part B, question came from these topics only, that is from critically endangered neuron, cell organelle, biodiversity, biofilm, that is bacterial biofilms, and cleavage, that is topic cleavage, types of egg from developmental biology. These question has been asked, and sugar conformation. They were giving a structure and they were asking you to identify which of the following sugar conformation structure is correct. So whenever you are referring to Leninger book, so draw the structure in your notebook. Okay. Next, there were a question from ecology, that is, there were a question asked from the prey predator relationship coming under species interactions. Okay. Next, nitrogen metabolism question has been asked from glycolysis pathway, and this GLUT, that is a glucose transporter, like from digestive system. They were asking how monosaccharides are absorbed and how they are exit this enterocytes. Okay. Next, there were questions starting from cell cycle regulation, so different stages of cell cycle, and when the chromosomes are visible, like that they were asking. Next, disease resistant gene, DNA markers, radioactive isotopes. So from this particular last unit, that is methods in biology, from this particular radio labeling technique, many question has been asked, that like detection measurement of different type of radioisotopes that are normally used in biology. So when you are going to prepare for next examination cycle, so please have an eye on radioisotopes used in biology field. Next, bacterial toxin, fertilization. So from developmental biology, they were asking a question with regarding to fertilization, which again an easy topic. I think many of you will be answering correct for this particular germination questions then. So that is with regarding to gametogenesis, fertilization and early development, that is cell surface molecule in sperm egg recognition in animal. They were question. Okay. Next, they were asking the ABC model and flower mutants. Okay. Next, thermoregulation, heat acclimatization question has been asked, and histone variants. This time many question has been asked regarding to histone variants and histone. Next, there were question asked from chlorophyll, cell culture assays, transcription factors. So if you study the structure of transcription factors from either from Pathfinder book, that well sufficient, and from pedigree chart, there were question has been asked there. Many question has been asked from this pedigree. Next, cluster of differentiation. So if you study the FACS technique, like FACS technique, then you can easily answer those questions that has been asked from the CD. Next, question has been asked from this ecology, that is population growth. Okay. So if you going to prepare next time, first complete the ecological principles. So this particular population growth and all asked under this population ecology. So even though you can't able to cover all the topic, just have an eye on this population ecology, species interactions, community ecology, and ecological succession in ecosystem ecology. Okay. Next, there were a question asked from respiration, biological species concepts, and biomes and forest types in India. And there were questions regarding to biosynthesis of small neurotransmitters and plant apomorphisms, repilosome in bacteria. So from bacterial replication, there were many questions asked: repilosome, replication, pore formation, replication initiation, like that. Many questions has been asked. Okay. Next, there were questions regarding to species richness, RK selection, coming under population ecology. So life history strategies, many questions has been asked. And regarding to electron transport chain, inhibitors of electron transport chain, genetic map, conjugation experiments like HFR mapping, autopolyploidy, and amino acylation of tRNA, many questions has been asked. This is regarding to part B, shift 1. Next, part C. So reciprocal translocation question has been asked. With regarding to first unit equation, that is Michaelis-Menten equation, many question has been asked. And then tDNA. So regarding to agrobacterial mediated gene transfer, many question has been asked from tDNA construct. Next, tetrapod limb development. So I think from developmental biology, tetrapod limb development question has been asked. 2 questions has been asked in shift 1 and 2 questions in shift 2. So at least have an eye on this tetrapod limb development in upcoming examination. Next, there were questions regarding to mammalian development, cell cycle regulation in eukaryote, denaturation, renaturation, kinetics of DNA, western blotting. Then there were questions asked regarding to cell adhesion protein, cell to cell communication, extracellular matrix, cell junction. Next, they had given a DNA diffraction pattern, and from that question has been asked. Next, Dixon-Platt enzyme inhibition question has been asked, and neuroglial cells and their function, and solute transport in plant like apoplast pathway, symplast pathway, transcellular pathway. Next, chromatin remodeling in eukaryote question has been asked, and from reverse genetic concepts like reverse genetic, forward genetic, there were 2 different genetic methods, and they were asking what is mean by random mutagenesis, targeted mutagenesis, and phenocopy. So even though you don't have time to study in depth, just know for what purposes random mutagenesis is employed, like that you should know. Next, every time from ECG only question has been asked, but this time from this EEG question has been asked, and they are asking the frequency waves and all. Okay. So next from this nucleotide excision repair question has been asked, and autonomous and conditional mode of specification in CL again question has been asked, and characteristics of different evolution process, like they were asking you to define parallelism, conversion, integration and hybridization. So whenever you are referring to few textbook, go to vocabulary section or glossary section, just read the definition statement. Next they were asking regulation of translation in prokaryote, photomorphogenetic response in plant, precursor, like they were asking a question from glycolysis and citric acid, and few molecules in glycolysis and citric acid will be acting as a precursor for few important biological pathway occurring in plant. Next our question regarding to biosynthesis of hemoglobin, heme, and cancer type and origin. So they were given a cancer and they were asking you to define the cancer or origin, from which particular part of the body the cancer is originating. Next our question regarding to biosynthesis of nucleotide, amino acylation of tRNA, interferons, hyposignaling pathway, and animal kingdom. Next floral induction protein, like flowering locus C, flowering locus D, flower transition genes. Okay, and they were asking us a question from structure of collagen, planaria regeneration, introns, pedigree, adaptive radiation, and how a DNA replication has been happening with the immense accuracy, which is responsible for accurately replicating a DNA without any mistake. Okay, so next there were questions regarding to ROS pathway and transcription initiation, and mutagens, two photon microscopy, factors that controlling a food intake. Next there were questions regarding to pituitary gland hormone, and they'll be asking you to identify odd one out like that. And there were a question asked from QB book immunology. There were many experiments that have been done with regarding to immunology, and if you refer only QB book, you are able to answer. So just if you take the printout of the experimental topics alone, and if you paste them, you can easily answer those questions. Same words has been taken from QB book, and they were asking few important questions. From QB book only, immunology experiment question has been asked. So one question has been asked from regarding to MHC and enzymes involved in cell cycle and the function of those enzymes. Next, they were involved in process of glycogenesis, glucogenesis, and abnormal expression of proteins in human cancer. Like what are the proteins that are abnormally expressed, and what is the consequence with reference to cancer? They were asking. And alpha helix, like they were asking what is the formula to calculate length of alpha helix, how many turns are present in alpha helix, and how many hydrogen bonds are present in alpha helix, like they were asking. And restriction enzyme digestion, they were asking application oriented. So if you're going to refer a restriction enzyme digestion, then if you're going to refer few textbook, at the last page they'll be asking you to do few problems, will be giving solved problems, you can work out those solved problems. Next, polar effect of mutation, auto regulation of blood they were question asked, and Bateman's principle, electrical and precordial burden, then gibberellic acid signaling. So this time regarding to plant hormone, only from gibberellic acid and from ABA only many question has been asked, both in shift 1 and shift 2. And there were question asked from insulators present in human gene, and what are the steps in signal transduction pathway. So step wise they are asking. Next, India's biodiversity also question has been asked. Okay, these are the list of topic from which question has been asked for shift 1. Next video I'll be definitely uploading a question that has been asked for shift 2 student. Thank you friends, thanks for watching this video.